All right, so this video real quick is just going to go over the three major neurotransmitters we care about and their entire cycle, essentially. So we're going to start with acetylcholine. I tend to draw acetylcholine in red for some reason. Doesn't matter, though. I'm not really trying to color coordinate anything in this lecture. I'm just trying to stay consistent with my own crazy thoughts. So acetylcholine is red, obviously. Glucose is made into pyruvate thanks to cellular respiration, yada, yada, yada. Eventually, this makes acetyl-CoA. This acetyl-CoA plus choline is turned into acetylcholine, right? I hope that makes some bit of sense. Acetyl-CoA plus choline equals acetylcholine. The enzyme responsible for this, we'll call this first enzyme, is going to be choline acetyl transferase. A lot of times you'll see this written as chat. Weird mnemonic I used to use is that your enzyme has a chat with acetyl and choline and convinces them to become acetylcholine. I don't know if that helps you. If not, just ignore me. But then it's trafficked into a synaptic vesicle by a vesicular protein. Remember, this is going to be an antiporter that's using the gradient of protons going out to drive acetylcholine in. This is going to be something called VACHT. This is a pretty common nomenclature. Let's say I was talking about glutamate. It would be V-glu-T or V-GABA-T or whatever it is. So this gets acetylcholine into, I should say, sorry, step back for one sec. V means vesicular. T stands for transporter. Acetylcholine is trafficked into the synaptic vesicle. This synaptic vesicle is released. So this is your presynaptic neuron we're talking about. Here's your postsynaptic neuron. Acetylcholine is released. And then there's a couple things it could do, right? So it could bind to a postsynaptic receptor, bind to some kind of acetylcholine receptor. We'll talk about all the different flavors of receptors we have, and it could be metabotropic, it could be inotropic, whatever it is. But again, we need some way to regulate the concentration of acetylcholine at the cleft, right? Because we don't want too much acetylcholine in the cleft, otherwise those receptors will get desensitized. So the method by which acetylcholine or cholinergic synapses do this is actually degradation. So we'll say number 2 here. The enzyme that degrades acetylcholine is going to be acetylcholine esterase. A lot of times you'll see this written as ACHE. That's acetylcholine esterase. That's the enzyme that degrades acetylcholine. And it degrades it into two products, right? Or two products. That's going to be acetate plus choline. There's actually a transporter. I don't really think I want you to know the name of this, but there's a transporter that gets choline back into the presynaptic neuron to then be combined with more acetyl-CoA to make acetylcholine again, and then it's released again, and the whole cycle continues in that manner. So the two enzymes we care about that you know are choline acetyltransferase. This is going to be the rate-limiting step in synthesis. Rate-limiting step in synthesis. And then this is going to be the removal mechanism. And again, this is going to be the degradation type of removal mechanism. So that's acetylcholine. So when we talk about glutamate, for me, glutamate just is blue. I don't know why. So here's a glutamatergic synapse. And So the precursor for a glutamatergic synapse is going to be a molecule called glutamine. So glutamine, which is also an amino acid, is going to be turned into glutamate by this first enzyme here. So that's glutamate. So here's our mature form of the neurotransmitter. This enzyme number 1, is going to be glutaminase. The way I remember that is that glutaminase makes it a maybe this doesn't work for this. I don't know how you're going to memorize that one. This is kind of annoying, but maybe if you know more about amino acid chemistry than I do, you might understand that better. But essentially, glutaminase is going to convert glutamine into glutamate. That's going to be trafficked into a vesicle by something called V-glut, which again is your vesicular glutamate transporter. Here's glutamate in there. That glutamate is going to be released. It's going to bind to receptors, be they metabotropic or inotropic, doesn't really matter. But then again, we need some way to remove this from the cleft, right? So we talked about glutaminase. We're going to call that our rate-limiting step in synthesis. Whereas the method by which we decrease the concentration of the cleft is actually going to be based on reuptake. So in this case, you have an astrocyte. Oftentimes it's called a perisynaptic astrocyte because it's peri means around. So it's the astrocyte or a glial cell that just basically envelops this synaptic cleft. And on that perisynaptic astrocyte, you're going to have something called EAAT. This is going to be an excitatory, so I'll just call this number 2. This is going to be your excitatory amino acid transporter. That's going to take glutamate from the synaptic cleft to move it into this astrocyte here. And then you have glutamate within the astrocyte. You don't want to release glutamate, so you want to have some way to get glutamate back to the presynaptic neuron. You don't want to release glutamate back into the cleft, though, because, again, the whole point of this excitatory amino acid transporter was to get it out of the cleft. So the astrocyte actually does something kind of interesting. It converts glutamate back into glutamine. And in this case, we actually have a third enzyme that we care about. So whereas this is the method for removal, is this excitatory amino acid transporter. Number 3 here is going to be glutamine synthetase. And this is a rare case in neuroscience where the name of the enzyme actually is a good name. This is an enzyme that makes glutamine. Then you can safely take glutamine and put it back into I drew that horribly. Take glutamine and put it back into the presynaptic terminal via some kind of transporter. I don't really care that you know what that is. But essentially, you're getting glutamine back into the presynaptic terminal. And if I had drawn this better from the get-go, I guess I should, let's see if I can just do this. It actually goes back into the synaptic cleft. And that transporter that brings it back in is actually within the synaptic cleft. It's kind of how this perisynaptic transport works. It's pretty local to this region of the astrocyte, so that's kind of a bad way to draw this. But essentially, you're putting glutamine back into the cleft where it doesn't have an activity, right? So glutamine does nothing in the synaptic cleft. So you're taking glutamate away, converting into something that's inactive in the cleft, and then putting it back in the cleft so that it can be safely trafficked back into the presynaptic neuron through this transporter that is specific to the presynaptic neuron, which is this transporter right here that I've drawn just horribly. Transporter right here. And then back to glutamine, so then you can then synthesize more glutamate within the presynaptic terminal. And then the last one I want to talk about is GABA. And for some reason, to me, GABA is purple, so that's where I'm going to draw it as. Oh, one thing I forgot to mention above here. Why would you think we call this an excitatory amino acid transporter? It might help you remember what kind of neurotransmitter glutamine is and what its overall effect is, right? So it's going to be mostly excitatory. It's going to be an amino acid neurotransmitter. So this transporter is going to take that excitatory amino acid, glutamate, and put it into the astrocyte. So for this last neurotransmitter, we have GABA. So the interesting thing about a GABAergic neuron, I talked about how dopaminergic neurons are dopaminergic because they don't contain those two enzymes that turn them into norepinephrine or epinephrine. Well, a GABAergic neuron, the only difference between a GABAergic neuron and a glutamatergic neuron is that whereas a glutamatergic neuron has just this glutaminase, a GABAergic neuron has glutaminase, right? Because it takes glutamine and turns it into glutamate. But then it takes this glutamate and turns it into GABA via something called glutamic acid decarboxylase. Oftentimes you'll see this written as GAD. So that turns glutamate into GABA. Then you have a vesicle that has something called a VIAAT. Again, this is just a stupid naming mechanism. They're saying vesicular inhibitory, right? So that's GABA. Amino acid, again, this helps you remember that GABA is an amino acid neurotransmitter. So this is an inhibitory amino acid transporter that's on the vesicle. So this is going to be what gets GABA into the synaptic vesicle. So here's GABA. This binding is going to lead to the release of GABA. And again, we have our three fates of a neurotransmitter. It could bind to a postsynaptic receptor. It probably could be degraded, but in this case, the removal mechanism is actually, again, reuptake. And this can actually occur via this perisynaptic, right? That word means around the synapse. So this could occur via the perisynaptic glial cell, which is an astrocyte. Or this neurotransmitter can actually just go straight back into the presynaptic terminal. So this transporter that I don't care, you know the name. Just for completeness sake, let's say that this is GAT, GABA transporter. And it exists both on the perisynaptic glial cell, this astrocyte, and the presynaptic terminal itself. So GABA can actually go straight back into the presynaptic terminal and be recycled pretty efficiently in that manner. So those are our three major neurotransmitters. For all the other ones, you have that huge table. And in the study guide I gave you, I actually blacked out all the things we don't need you to know. I hope it makes it seem a lot more manageable. In terms of precursors and rate-limiting steps and removal mechanisms, all we care about that you know is those things for acetylcholine, glutamate, and GABA. Like, we don't care if you know the precursors for any of the other neurotransmitters, or we don't care if you know the thing that eventually makes them. And we definitely don't care what type of vesicle for any neurotransmitters, even the ones we talked about here, because I don't really know what they're trying to get at with that. That's probably more of a histology thing than anything else. Definitely know the postsynaptic effects, but know that those postsynaptic effects are mediated by the receptor, not the neurotransmitter themselves. Next we're going to talk about all the different slew of receptors we have, ionotropic and metabotropic.